The nervous system. Ta-da! Every time you move a muscle, every time you think a thought, your nerves are hard at work. They're processing information, receiving signals, deciding what to do with those signals, dispatching new messages. Nerve cells communicate directly with other muscle cells. Nerve cells communicate with each other up your spinal cord into your brain. Some nerve cells are involved solely with information. Some are only about secreting certain proteins and chemicals. And different animals have different sorts of complexity to their nervous systems. So here's a cross-section of the brain. Here's a nice image of the brain where you can see the midbrain. Here's showing you some neurons conducting, and here's a typical model of a neuron. And then the scarecrow. What's that? What's he doing there? Remember how he wanted a brain? And then at the end, when he supposedly thinks he really already had a brain, and he says the Pythagorean theorem, he says it wrong. So there's your trivia for Wizard of Oz for today. You can go back and listen to it. I forget what he says, but I think he describes an isosceles triangle instead of, or a right triangle instead of an equilateral triangle, something like that. Oh wait, there's a hearing button here. Does he say something? Oh no, media not found. Oh, maybe it was going to have him say it. Sorry. Okay, so why do animals need a nervous system? Alright, there's that penguin. And I don't know why he's saying think about the bunny, but anyway, think about the bunny for a moment. Here's a hawk. Think about the relationship between the two of them. Alright, and here, I don't know how well you can see it, but you can see where there were bunny prints in the snow, and clearly a hawk came down and swooped up the bunny. So the idea is that you need a nervous system so that you are able to survive. The bunny didn't make it, but hey, the hawk had a good meal out of it. So what characteristics do animals need in a nervous system? Well, especially in the case of both of these animals, fast. They need to be able to respond fast. Bunny should be able to run fast. Hawk should be able to plunge fast to catch food. Okay, needs to be accurate, and needs to be able to reset quickly. So if it can't reset and it's overfired, then you're not going to be able to respond again to the next stimuli. So here's their neuron, the basic nerve cell. Obviously, not a cell that's a big blob of a circle, like we show you in basic bio. So we're going to talk a little bit about the parts here. All right, this is the cell body, and in the cell body is where we have the typical parts of the cell, the organelle, the nucleus, endoplasmic reticulum, ribosomes, etc. Then we have dendrites, which do what they look like they're going to do. Right? They're going to spread out to carry information. Here's an axon conducting a signal in this direction, and we're going to talk about that quite a bit. Okay, and over top of this is a myelin sheath. Myelin is kind of a fatty substance. And then at the end, we have a synaptic terminal, terminal meaning end, and a synapse. And I'm going to take the next couple slides to go over a little bit of this with you, and then we'll kind of hit the rest of these details as you learn about nerve conduction. So definitely spend some time learning just what parts are called what, because it'll make things much easier moving forward. Okay, so structure fits function in this case, obviously. We have many entry points for signal and one path out. In other words, as I was saying, these dendrites look like they're reaching out for a signal, and then we're going to transmit a new signal out in this one direction. Okay, so the dendrites, just to talk about them for a second, I just threw this in there, that's why there's no images on it, just so you have a good definition, a little bit more than what your book says. That they're tree-like extensions at the beginning of a neuron that help increase the surface area of the cell body and are covered with synapses. They transmit electrical stimulation to the soma, which is the cell body. They're short and highly branched. Most of the neurons have dendrites, and it transmits information to the cell body or soma. All right, here. The cell body, also known as the soma, contains all the organelles and is the site of protein synthesis. So maybe you can kind of make this, oopsie, back. Make this bigger for yourself so you can kind of appreciate that. So even though this is a big, funky looking cell, all that good stuff that we're used to thinking about in cells are happening in the cell body, which is also known as the soma. Remember that soma means body. Alright, cool stuff about neurons. Okay, they're very specialized. The longest cell is a blue whale neuron, which can be between 10 and 30 meters. Okay, a giraffe axon, remember what an axon, what part of a neuron that is, could be about 5 meters. And a human neuron can be about 1 to 2 meters. And this is a little bit debatable, but what they're talking about in which neuron of ours is 1 to 2 meters, they're talking about the nerve that runs from the base of our spinal cord down our legs. But it's kind of segmented, so 1 to 2 meters is kind of not the best way of thinking about it. There are some fibers in there that seem to literally run that long, but it's unlikely that there's still indeed one intact neuron. But anyway, that controversy is not that important. Okay, so the nervous system allows for 1 millisecond response time, and sometimes faster than that. Okay, so transmission of the signal. That's what we're going to be spending a bunch of time talking about here. Thinking about dominoes, when you set up a bunch of dominoes and you knock over the first one and it's like, and the whole thing comes down. Alright, so knocking down a line of dominoes by tipping the first one triggers the signal. And we have a starting the signal, and then we have what's called propagate the signal, propagation. Do the dominoes move down the line? No, just a wave through them. I don't know why they say it quite that way. We're going to come back to this. The dominoes is not a great way of explaining this anyway. And then resetting the system. So before you can do it again, you have to set it back up and reset the axon. So the idea is that these are the 3 key words, that you start a signal, you propagate the signal, moving it down the neuron, and then reset the system. All right, let's get to the real neuron stuff, because the domino is making me annoyed. All right. Okay, so there are protein channels, and we've talked a lot about protein channels, and now you're going to really see how important they are, and that when we talk about diseases of the nervous system, that these are the parts of the nervous system that we think we have a chance of treating with medication as affecting protein channels. So once one protein channel opens, the rest open in succession. It's a all or nothing. A wave action travels along a neuron, and you have to reset the channels so the neuron can react again. All right, I think the next slide has the animation on it. All right. So cells are surrounded by charged ions, and if you look at this picture, even though we have some positive and negative ions inside and outside, we have a lot more negative ions, anions, within the cell, such as chlorine, and we have a lot of positive cations in the extracellular fluid, the fluid around the cell. In this case, mostly sodium. And I'm sure you remember the sodium-potassium pump too. All right, so there's a charge difference between the inside and the outside of the membrane. And that charge difference is really, really important when we're talking about neurons. And the other thing that they're pointing out here is that there are channels kind of leaking the potassium out. So to some degree, the membrane is semi-permeable to the potassium. We'll talk a little bit more about that in a couple slides. Okay, so cells have voltage because of the opposite charges that are created. On the outside of the cell, positive, inside the cell, negative. This creates an imbalance. That imbalance is really cool for us. The positively charged ions repel each other, as do the negatively charged ions. They want to flow down their concentration gradient and mix together evenly. This means that there's energy stored here like a dammed up river. Voltage, you've probably learned about voltage somewhere along the line, is a measurement of stored electrical energy. Like when you see a sign that says danger high voltage, that's dangerous because there's a lot of electrical energy, right? And so we can talk about cells in terms of voltage too. So the membrane is polarized. It has a charge, negative inside, positive outside. That's important to remember. Negative inside, positive outside. I don't know how you remember that. Well, we'll have to come up with a way. Alright, so it has a charge gradient, and it stores energy like a battery. And we're talking about electrical energy. So how are we going to measure that? Alright, so voltage is the difference in concentration of charges. So the difference between the positive charges and the negative charges. The positives are the hole you leave behind when you move an electron. Oops, back it up. I have such a sensitive mouse. Alright, the original experiments on this were all done on giant squid neurons, which I think is in your book somewhere. So let's start from the beginning here. If we're talking about a neuron, okay, and we went over the parts, here's the axon, and we're looking at a chunk of the axon here, okay? An unstimulated neuron, a neuron just hanging out, has a resting potential, remember potential energy like a ball at the top of a stairs, of minus 70 millivolts. And this is a really key number. So an unstimulated neuron hanging out has a resting potential energy of minus 70 millivolts. Oops. There we go. So this number is determined by figuring out the difference between the charges on the inside and the charges on the outside created from the sodium and potassium. And they explain exactly where all of those numbers come from in your book on page 1016. So we can go over that in class a little bit, but it helps if you kind of check that out first. Alright, so how does a nerve impulse travel along this? Now remember here, we're looking at a chunk of the axon, a chunk of the neuron. So stimulus, the nerve is stimulated. So instead of resting potential, we're going to reach threshold potential. And sodium channels are going to open. And sodium ions are going to go into the cell. Those positive ions are going to move inward. And that charges charges then reverse at that point on the neuron. Right? So why here? So once it becomes positive inside, it becomes negative outside, because we're kind of switching around the polarity when we move some sodium inside. So the cell becomes depolarized. Okay, the first domino goes down. All right, so stimulus happens. Sodium channel opens. The positive charge diffuses into the cell. Right away we get a flipperoo then. So we have a positive charge inside, negative outside, and the cell becomes depolarized right here. And now that action is going to start moving down the axon. Okay? So if you remember, we were talking about how thoughts, how everything becomes a part of this chemical, electrical signaling process. Well, that's what we're learning about. And here's the beginning of that process. Sodium rushing in, depolarizing this part of the axon. All right, so how is it going to travel? Well, it travels in a wave. Nerve impulses travel down the neuron. So remember, what started over here, and now it's going this way. And it's always going to go in that way. So a change in the charge opens the next sodium gate down the line. So we started with this gate here, and then it was this gate here, and then it was this gate here. And if you notice, now we have positive ions inside the cell and negative outside because we're depolarizing the cell. Right? And so these channels are called voltage-gated channels. And here's a little picture of them. It kind of looks like all the other kind of membrane channels we've shown you before. We have a little animation of that here at the end, I think. So sodium ions continue to diffuse into the cell once this stimulus has occurred. And waves move down the neuron, and we start to reach the action potential. And don't panic yet, we're going to talk all about that. All right, this movie is almost 18 minutes long. So I'm going to pause right here before we get to the rest of action potential. So this is the end of part 1. Woohoo! Okay. Okay, so this is nervous system part 2. Although it kind of looks like it's picking up from yesterday. I'm not sure if that's true or not. Anyway, just to remind you, we're talking about that cells have voltage because there's an opposite charge building up, a negative charge on the inside of the cell and a positive charge on the outside. I was looking at another person's video, and they called the neuron a salty banana, that there was a lot of sodium ions on the outside and potassium ions on the inside, but there was a lot more positive sodium ions on the outside than any positive ions on the inside. So we talked about the membrane being polarized, and that creates a gradient between the charges and stored energy like a battery. And if you remember, it was negative 70 millivolts as a resting potential. So when a neuron is not being stimulated, it has potential energy, and that potential energy has a number attached to it of minus 70 millivolts. So, how does a nerve impulse travel? First, you have a stimulus that causes a difference in this charge. The sodium is going on into the cell, into the neuron, so that a threshold potential is reached. Because remember that there's a potential sitting there, and there has to be enough stimulus for the whole thing to fire. So when that happens, the sodium channels open, sodium diffuses into the cell, and the charges reverse at that point on the neuron. And the cell becomes depolarized. So this is the first chain in the link of dominoes, as the penguin will tell you. So then it's going to move down. So a wave is the nerve impulse moving down. And you can see the changes in charges behind that impulse. So the next sodium gate goes down the line, then the next one, then the next one, so that we have more positive charge on the inside of the neuron. Okay, and these are voltage-gated channels, because remember we're talking about voltage here, electrical charge. So here is a picture from your textbook showing channel closed and channel open. The sodium ions are going to continue to diffuse into the cell, and the wave is going to move down the neuron, and we call that the action potential. So we talked about the stimulus potential, the threshold, and the action potential so far. Okay, so once we've got a good stimulus going that reached the threshold, it's going to cause firing all the way down. All right, so how does a nerve impulse travel? Continuing, reset. The second wave travels down the neuron. So remember, this is where we started, that this is the sodium channels opening and we're getting this reversal of charge. Well, we need for that neuron to return to its base state. So it's going to reset by potassium channels opening. So we want to get back to this whole original situation of negative on the inside, positive on the outside. So now potassium channels open. All right, potassium channels open up more slowly than sodium channels. So the potassium ions diffuse out of the cell and the charges reverse back to where they were originally. So we end up with negative inside and positive outside, back to the way we like it. So we're quickly setting the dominoes back up, waiting for another impulse to come along and fire them all again. Alright, so the combined waves travel down a neuron. So you've got this impulse here, the stimulus, moving down the neuron, and a little bit behind it is the potassium, resetting the neuron to its original state. And the signal only moves in that one direction. Okay. So, action potential propagation. Well, we have the nerve impulse or action potential that we've been talking about. For example, from your brain to your fingertip, that idea, okay, or that motion can happen in milliseconds, okay, in the blink of an eye. Right, voltage-gated channels, ion channels open and close in response to changes in charge across the membrane. And that's what we've been talking about here. It's just explained a little bit deeper. The sodium channels open quickly, and they have to. If you think about how fast your nervous system needs to respond to a stimulus, the sodium channels need to open quickly. Resetting can happen more slowly, but the impulse needs to happen really, really, really fast. All right, the potassium channels go more slowly and close slowly, and so we slowly return to the original state of the neuron. All right, structure and function, of course, of course. So here's a picture trying to show you that resetting. Okay, after firing a neuron, it has to reset itself. So sodium has to move back out, and oh, this picture is a little bit crazy. All right, potassium needs to move back in, and both are moving against the concentration gradients. Okay, so just to be clear, from here that way is still the wave of the nerve that we were talking about. Here's the stimulus, and here's it's trying to return to normal. And back here, we're having a normal neuron. All right, so if we're moving against a concentration gradient, is that passive or active transport? Yes, it's active. So what do we need? A pump. And we talked about this pump before when we talked about the cell membrane, but we didn't go into much detail, but now we will. All right, so the sodium-potassium pump we talked about before, and here it is. Okay, pumping sodium out of the cell and potassium in to return a neuron back to its original state. Okay, a lot of work to do here. All right, so the sodium-potassium pump. the nerve resetting itself. Active transport in the membrane. Of course, since it's active transport, it requires energy. Ta-da! ATP. Oh no, the sound for ATP was Alright, so the exchange rate for the potassium pump, and we've talked about this before, is 3 sodium ions are pumped out. And how many are pumped in? How many potassium? 2 potassium are pumped in. So this resets the charge across the membrane. Is the charge neutral? No. Okay, we said that the neuron always has a charge, and its resting charge is minus 70 millivolts. Right, and it's a lot of ATP to do this. Right, here's a picture showing you the inside of the cell and the outside of the cell. Excuse my yawning, it's actually 5:30 in the morning while I'm finishing this up for you guys. So the sodium-potassium pumps are one of the main drains on ATP production in your body because your brain is a very energy expensive organ to run. So this pump you've seen before, that it's perfectly shaped. It's, you know, a chunk of protein shaped just for the exchange of sodium and potassium, but it requires a bunch of ATP for it to work. Alright, so here, the neuron being all reset and ready to fire again. By now you're wondering that you thought the nervous system was going to be all about the brain, and we will get there, but first you have to understand how neurons fire. That's why we're starting here, even though your book goes in a kind of different order. Right, so here's a neuron perfectly reset back to its resting potential. Okay, so this graph is a really common graph to see. I'm going to just pop up all the parts of it here. Okay, and where is this in your book? This is on page 1018 in your book. And the picture in your book, it would be easier if you had that open while we talk about it, because it shows you unstimulated and then stimulated. So can I make this larger? I suppose not. But what we're trying to show you here is that a nerve is going to be at its resting potential, okay, and then it's going to be stimulated. And when it hits the threshold of stimulation, it's going to fire. And that's going to be leading to the depolarization that we just talked about of the sodium and potassium. And after that, it's going to repolarize and return to its resting potential. And that's all that's to it. The reason we show it in this graph is it helps you understand when something is wrong with a nerve, because it doesn't quite work this way. Some nerves can be functioning where the threshold is set too low and nerves fire too quickly, or up higher where they fire too slowly. So the key terms to know here are the resting potential, the threshold potential, depolarization, then the channels, sodium channels closed, potassium channels open. We repolarize so that we have that charge across the membrane. And then undershoot is the part where the potassium channels are closing slowly. All right, kind of simple once you think about it. So we have a nerve hanging out, then it starts to be stimulated, and when that stimulation hits a threshold, it really fires. And we have a massive, fast depolarization, and sodium rushes in. And when that's reached a peak for that neuron, it starts to go the other way, where we start to repolarize and potassium flows out. Then it's going to dip a little bit under as the potassium channels close slowly and eventually return to resting potential. Okay, so talking a little bit more about the neuron itself here, the myelin sheath. Okay, axons down here are coated with cells called Schwann cells. and they are fatty, and they insulate the axon. And fat is a great conductor, so it speeds up the signal. All right, so this, you see that this is broken into little like beads on a string. So the signal hops from node to node along the axon. And it's a saltatory conduction. And saltatory is right on your book on page 1021, and it means this idea of jumping from node to node. Okay, so there's the little jumperoo. And what's jumping is the signal. Okay, and this is the speed of that signal. This book, or this picture comes right from your book on page 1021 also. So let's look at it a little bit closer here. Okay, so we're having, still talking about action potential, we're looking at the axon, the myelin sheath, and saltatory conduction, leaping, okay, from node to node over along the Schwann cells. These are the Schwann cells. These little spots in between are called the node of Renvier, okay, and that's a region of depolarization. All right, so signal, jumping, jumping, jumping. We're going to get to how they communicate with each other too. All right, so in multiple sclerosis, a disease of the nervous system, the immune system attacks the myelin sheath. There's more to it than that, but that's a basic part of what can happen. So that conduction along neurons is not regular, and you have a loss of the signal. Let's talk about the end of the axon, communicating from nerve to nerve. Okay, so the space between the axon of one nerve, okay, and the dendrites of another is called a synapse. You've heard that term before. Okay, so it's this little space in between, and there's definitely a gap there, and that gap is called the synaptic gap. So the penguin's going to ask you, okay, how does the wave jump the gap? So an impulse has to jump that synapse. So signal's got to be strong enough to jump along here and then cross the synaptic gap, the junction between neurons. So it has to jump quickly from one cell to another if we want to get a signal from your finger to your brain or the other way. So here's a closer look at a chemical synapse. And this picture is also from your book. Not completely exactly the same, but on page 1022. It was talking about chemical synapse. So how chemicals go across the space. So we've talked before about calcium being a very important ion throughout your body, and it's involved in a ton of processes. And so here calcium is very important in the synaptic gap. All right, so here's your axon, okay, and here's the end of the axon terminal, and the potential, okay, that leaped along is coming here. All right, and what's inside these vesicles, okay, these are called synaptic vesicles, and they contain neurotransmitters, chemical signals, in this case, acetylcholine. So for that acetylcholine that this neuron is transmitting to go across the synaptic gap into, in this case, a muscle cell, all right, the vesicles are going to have to reach the end of the cell, open up. These neurotransmitters are going to have to go across the space and go into the channels in through these little receptor proteins in the muscle cell to get in there. Okay, and then, all right, so the action potential depolarizes the membrane, opens the calcium channels, the neurotransmitter vesicles fuse with the membrane, so they're up there, just like we talked about exocytosis. They release their contents, the neurotransmitter. The neurotransmitters go across the gap and go into the muscle cell via diffusion. So a neurotransmitter binds with a protein receptor and heads on in to affect that muscle cell. Right, these are ion-gated channels opening. All right, so we switch from an electrical signal to a chemical signal. And I know I have shared many times in class that your nervous system is both electrical and chemical, and that's super important. Okay, so the neurotransmitter degraded or reabsorbed. I don't know exactly what that means, because it's either, I guess they're talking about when it doesn't go across, because it is going to go across, and what doesn't go across is going to be reabsorbed or degraded in the space. We'll talk about what it does to the muscle cell in a second. Okay, so a postsynaptic neuron, okay, triggers a nerve impulse in the next cell. So this is not our original cell, this is the second cell. All right, you remember what this depolarization looked like in the first cell, so here it is in the second cell. The chemical signals the ion-gated channels to open, and we start, in this case we've got the acetylcholine. Okay, sodium diffuses into the cell, potassium diffuses out of the cell, and we switch back to a voltage-gated channel. So here we go again. So let's talk about the neurotransmitters, and these you do have to know. Acetylcholine transmits a signal to skeletal muscle. So what you saw there was where a motor neuron, okay, a neuron involved with movement, transmits a signal to skeletal muscle. So why does skeletal muscle move? It moves because it receives acetylcholine from a nerve. Okay, epinephrine and norepinephrine. You've learned the term adrenaline somewhere, but what adrenaline really is called epinephrine. And you know that adrenaline is like your fight or flight reaction. So that's what we're going to call it here too. So our fight or flight response is controlled by epinephrine and norepinephrine. Dopamine, okay, has a lot of different functions in the brain, and it's all over the brain. And it affects sleep, mood, attention, learning, and when you don't have dopamine, you have Parkinson's disease. So Parkinson's disease is characterized by a lot of abnormal movements that are related to not having dopamine. If you have too much dopamine, you can have schizophrenia. All right. Another big neurotransmitter is serotonin, which is also widespread in the brain. It also affects sleep, mood, attention, and learning. If a lot of antidepressants are currently aimed at serotonin, they are called serotonin reuptake inhibitors. So in the synaptic gap, if you are depressed, serotonin doesn't go completely from one neuron to another. It gets reabsorbed by the original neuron. And so antidepressants force the serotonin across the synaptic gap, so that aids depression. Let's see, the notes here that you'll see when you are looking at the actual PowerPoint, when it's mailed to you, talk about GABA too. I guess I must continue on the next page, so hang on. Oh, this doesn't talk about GABA. Anyway, we'll go over the rest of those neurotransmitters in class. But needless to say, the ones in your book are the ones that you need to know. They're on page 1024. Okay, so neurotransmitters are considered a weak point in the nervous system because any substance that affects them or mimics them affects the way nerves function. For example, gases, such as nitrous oxide and carbon monoxide, which are poisonous, can affect them. Mood-altering drugs, stimulants, such as amphetamines, caffeine, nicotine, depressants, quaaludes and barbiturates, hallucinogenic drugs, LSD, peyote, and then selective serotonin reuptake inhibitors, like I was talking about earlier, the antidepressants, Prozac, Zoloft, and Paxil, all of these affect neurotransmitters, as do poisons. Oh, we are going to go into more detail here. Okay. Okay. Just from looking at this word, acetylcholine esterase, you kind of know it's an enzyme because it's got ASE, and it's going to break down acetylcholine, neurotransmitter. So, things that inhibit acetylcholine esterase are neurotoxins because acetylcholine is essential for life. Okay, so some of the neurotoxins that do this are snake venom, sarin, and insecticides. So if you're a landscaper, say, and you are too close and inhale a bunch of insecticides, you can be at risk for being poisoned by that. So here's the protein, the enzyme, acetylcholinesterase, and here's snake venom, the toxin from snake venom, binding to it and blocking the active site. So acetylcholinesterase is considered a weak point in the nervous system because poisons and toxins that attack the enzyme cause acetylcholine to accumulate in the nervous system. If acetylcholine builds up, you're going to paralyze muscles. So if you get attacked by a snake, you're going to be paralyzed. Acetylcholine esterase, the enzyme, is found in the synapse between nerve cells and muscle cells. So it's just hanging out there and springs into action after a signal is passed. So when the signal happens, acetylcholine breaks into acetic acid and choline, stopping the signal, allowing the pieces to be recycled and built into a new neurotransmitter all over again, the recycling. So one of the questions that we could ask here, that I'm going to ask you, is acetylcholinesterase toxin a competitive or non-competitive inhibitor? Is acetylcholinesterase toxin a competitive or non-competitive inhibitor? Alright, so there's the active site, here's the neurotoxin. Okay, questions to ponder. Why are axons so long? Why have synapses at all? How do mind-altering drugs work? Do plants have a nervous system? Do they need one? Ponder this. Any questions? Well, sure, I have some questions for you that you're going to answer. And here, I'm going to write down, they're actually the ones from the book, so hang on one second. Alright, so to make things simple, this is your homework to go with this PowerPoint. I realize that this is some complex material, so I'd like you to review it in the book as well. So I want you to answer these questions from these pages, and obviously you can check your answers in the back of the book. So I would like you to answer them, try to answer them on your own, and then make the corrections from the back of the book. Additionally, please make flashcards for studying the neurotransmitters, and then draw and label the action potential, because if you spend a little time drawing it, it's going to make a lot more sense to you. So obviously in this video we didn't talk about the brain and the spinal nerves and the anatomy yet, because that will be in another video. Thank you very much.